Today we'll read chapter 3 of class 11 biology, that is plant kingdom. In the previous chapter we looked at the broad classification of living organism under the system proposed by Whittaker in 1969, wherein he suggested the 5 kingdom classification, that is Monera, Protista, Fungi, Animalia and Plantae. In this chapter we will deal in detail with the further classification within the kingdom Plantae, popularly known as the plant kingdom. We must stress here that our understanding of the plant kingdom has changed over time. Fungi and members of the Monera and Protista having cell walls have now been excluded from Plantae, though earlier classification put them in the same kingdom. So, the cyanobacteria that are also referred to as a blue-green algae are not algae anymore. In this chapter, we will describe Plantae under algae, bryophytes, pteridophytes, gymnosperm and angiosperms. Let us also look at classification within angiosperm to understand some of the concerns that influence the classification system. The earliest system of classification used only gross superficial morphological characters such as habitat, color, number and shape of the leaves, etc. They were based mainly on vegetative characters or on the androecium structure system given by Linnaeus. Such systems were artificial. They separated the closely related species since they were based on the few characteristics. Also, the artificial system gave equal weightage to vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative characters are more easily affected by environment. As against this, natural classification systems developed which were based on natural affinities among the organisms and consider not only the external feature but also internal features like ultrastructure, anatomy, embryology and phytochemistry. Such a classification of flowering plant was given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification system based on the evolutionary relationship between the various organisms are acceptable. This assumes that organisms belonging to the same taxa have a common ancestor. We now use information from many other sources too to help resolve difficulties in classification. These become more important when there is no supporting fossil evidence. Numerical taxonomy, which is now easily carried out using computer, is based on all observable characteristics. Number and codes are assigned to all the characters and the data are then processed. In this way, each character is given equal importance and at the same time hundreds of characters can be considered. Cytotaxonomy, that is based on cytological information like chromosome number, structure, behavior and chemotaxonomy that uses the chemical constituents of the plant to resolve confusions, are also used by taxonomists these days. Algae. Algae are chlorophyll bearing simple thalloid autotrophic and largely aquatic both freshwater and marine organisms. They occur in varieties of other habitat like moist stones, soil and wood. Some of them also occurs in association with the fungi known as lichen and animals, examples on sloth bear. The form and size of algae is highly variable. The size ranges from a microscopic unicellular form like Chlamydomonas to colonial form like Volvox and to the filamentous form like Eulothrix and Spirogyra. A few of the marine forms such as kelps form massive plant bodies. The algae reproduce by vegetative, asexual and sexual methods. Vegetative reproduction is by fragmentation. Each fragment develops into a thallus. Asexual reproduction is by the production of different types of spores, the most common being the zoospores. They are flagellated, that is motile, and on germination gives rise to new plants. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size as in the case of Chlamydomonas, or non-flagellated, non-motile but similar in size as in Spirogyra. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size, as in some of the species of Chlamydomonas, is termed as anisogamous. Fusion between one large non-motile, that is static, female gametes and a smaller motile male gamete is termed as oogamous. Example, volvox, fucus, etc. Algae are useful to man in varieties of ways. At least a half of the total carbon dioxide fixation on the earth is carried out by algae through photosynthesis. Being photosynthetic, they increases the level of dissolved oxygen in their immediate environment. They are of paramount importance as a primary producer of energy-rich compound which form the basis of a food cycle of all aquatic animals. Many species of Porphyra, Laminaria and Sargassum are among the 70 species of marine algae used as a food. Certain marine brown and red algae produces large amount of hydrocolloid, that is water holding substances. Example, algin, that is produced by brown algae, and keratin, which are produced by red algae, are used commercially. Agar, one of the commercial products obtained from Gelidium and Gracilaria, are used to grow microbes and in preparation of ice creams and jellies. Chlorella and spirulina are unicellular algae rich in protein and are used as a food supplement even by the space travelers. The algae are divided into three main classes: Chlorophyceae, Phaeophyceae and Rhodophyceae. First one is Chlorophyceae. The member of Chlorophyceae are commonly called green algae. The plant body may be unicellular, colonial or filamentous. They are usually grass green due to the dominance of pigment chlorophyll A and B. The pigments are localized in definite chloroplast. The chloroplast may be discoid, plate-like, reticulate, cup-shaped, spiral or ribbon-shaped in different species. Most of the members have one or more storage bodies called pyrenoids located in the chloroplast. Pyrenoids contains protein besides starch. Some algae may store food in the form of oil droplet. Green algae usually have a rigid cell wall made of an inner layer of cellulose and an outer layer of pectose. Vegetative reproduction usually takes place by fragmentation or by formation of different types of spores. Asexual reproduction is by flagellated zoospores produced in zoosporangia. The sexual reproduction shows considerable variation in the type and formation of sex cells, and it may be isogamous, anisogamous or oogamous. Some commonly found green algae are Chlamydomonas, Volvox, Eulothrix, Spirogyra and Cara. Second type is Phaeophyceae. The member of Phaeophyceae or brown algae are found primarily in marine habitat. They show great variation in size and form. They range from simple branched filamentous form like Ectocarpus to profusely branched form as represented by kelps, which may reach a height of 100 meters. They possess chlorophyll A, C, carotenoid and xanthophylls. They vary in color from olive green to various shades of brown depending upon the amount of xanthophyll pigment fucoxanthin present in them. Food is stored as a complex carbohydrate which may be in the form of laminarin or mannitol. The vegetative cells have a cellulosic wall usually covered on the outside by a gelatinous coating of algin. The protoplast contains, in addition to plastid, a centrally located vacuoles and a nucleus. The plant body is usually attached to the substratum by a holdfast and has a stack, the stipe, and a leaf-like photosynthetic organ, the frond. The vegetative reproduction takes place by fragmentation. Asexual reproduction in most of brown algae is by biflagellated zoospore that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous or oogamous. Union of gametes may take place in water or within the oogonium, that is oogamous species. The gametes are pyriform, that is pear-shaped, and bear two laterally attached flagella. The common forms are Ectocarpus, Dictyota, Laminaria, Sargassum and Fucus. Third one is rhodophyceae. The members of rhodophyceae are commonly called red algae because of the predominance of red pigment or phycoerythrin in their body. Majority of the red algae are marine with a greater concentration found in the warmer area. They occur in both well-lighted region close to the surface of water and also at a great depth in oceans where relatively little light penetrates. The red thalli of the most of the red algae are multicellular. Some of them have complex body organization. The food is stored as a floridine starch which is very similar to amylopectin and glycogen in structure. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by non-motile gametes. Sexual reproduction is oogamous and accompanied by complex post-fertilization development. The common members are Polysiphonia, Porphyra, Gracilaria and Gelidium. Next is bryophytes. Bryophytes includes the various mosses and liverworts that are found commonly growing in moist shaded areas in the hills. Bryophytes are also called amphibians of the plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid and shaded localities. They play an important role in plant succession on bare rocks or soil. The plant body of bryophytes is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by unicellular or multicellular rhizoids. They lack true roots, stem or leaves. They may possess root-like, leaf-like or stem-like structure. The main plant body of the bryophytes is haploid. It produces gametes, hence is called as gametophyte. The sex organs in bryophytes are multicellular. The male sex organ is called as antheridium. They produces biflagellated antherozoids. The female sex organ called archegonium is flask-shaped and produces a single egg. The antherozoids are released into water where they come in contact with the archegonium. An antherozoid fuses with the egg to produce a zygote. Zygotes do not undergo reduction division immediately. They produce a multicellular body called a sporophyte. The sporophyte is not free living but attached to the photosynthetic gametophyte and derives nourishment from it. Some cells of the sporophyte undergo reduction division, that is meiosis, to produce haploid spores. These spores germinate to produce gametophyte. Bryophytes in general are of little economic importance, but some mosses provide a food for herbaceous mammals, birds and other animals. Species of sphagnum, a moss, provide peat that have long been used as a fuel, and because of their capacity to hold water as a packing material for transshipment of the living material. Mosses along with the lichens are the first organisms to colonize rock and hence are of great ecological importance. They decompose rocks, making the substrate suitable for the growth of higher plants. Since mosses form dense mats on the soil, they reduces the impact of falling rain and prevents the soil erosion. The bryophytes are divided into liverworts and mosses. Liverworts. The liverworts grow usually in moist, shady habitats such as banks of streams, marshy ground, damp soil, bark of trees, and deep in the woods. The plant body of the liverworts is thalloid, example Markantia. The thallus is dorsoventral and closely appressed to the substrate. The leafy members have tiny leaf-like appendages in two rows on the stem-like structure. Asexual reproduction in liverworts takes place by fragmentation of thalli or by the formation of specialized structure called as gemmae, single gemma. Gemmae are green multicellular asexual bud which develops in small receptacles called gemma cups located on the thalli. The gemmae becomes detached from the parent body and germinate to form a new individual. During sexual reproduction, male and female sex organs are produced either on the same or on different thalli. The sporophyte is differentiated into a foot, seta, and capsule. After meiosis, spores are produced within the capsule. These spores germinate to form free-living gametophyte. Next is mosses. The predominant stage of the life cycle of a moss is a gametophyte, which consisting of two stages. The first stage is the protonema stage, which develop directly from a spore. It is a creeping, green, branched and frequently filamentous stage. The second stage is a leafy stage, which develop from the secondary protonema as a lateral bud. They consisting of upright, slender axis bearing spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoids. This stage bears sex organs. Vegetative reproduction in mosses is by fragmentation and budding in the secondary protonema. In sexual reproduction, the sex organs antheridia and archegonia are produced at the apex of the leafy shoots. After fertilization, the zygote develops into a sporophyte consisting of a foot, seta and a capsule. The sporophyte in mosses is more elaborate than that in liverworts. The capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanism of spore dispersal. Common examples of mosses are Funaria, Polytrichum and Sphagnum. Next is pteridophytes. The pteridophytes includes horsetail and ferns. Pteridophytes are used for medicinal purpose and as a soil binders. They are also frequently grown as ornamentals. Evolutionarily, they are the first terrestrial plants to possess vascular tissue, that is xylem and phloem. You shall study more about these tissue in chapter 6. The pteridophytes are found in cool, damp, shady places, though some may flourish well in sandy soil conditions. You may recall that in bryophytes, the dominant phase in the life cycle is a gametophytic plant body. However, in pteridophytes, the main plant body is a sporophyte, which is differentiated into true root, stem and leaves. These organs possess well differentiated vascular tissues. The leaves in Pteridophyta are small, that is microphytes as in Selaginella, or large, that is macrophytes as in ferns. The sporophytes bear sporangia that are subtended by leaf-like appendages called sporophytes. In some cases, sporophytes may form a distinct compact structure called as strobili or cones, like Selaginella or Equisetum. The sporangia produces spore by meiosis in spore mother cells. The spores germinate to gives rise to inconspicuous, small but multicellular free-living, mostly photosynthetic thalloid gametophytes called as prothallus. These gametophytes require cool, damp, shady place to grow. Because of this specific restricted requirement and the need for the water for fertilization, the spread of the living pteridophyte is limited and restricted to a narrow geographical region. The gametophyte bears male and female sex organ called as antheridia and archegonia respectively. Water is required for transfer of antherozooids, the male gametes released from the antheridia, to the mouth of the archegonium. Fusion of male gametes with the egg present in the archegonium results in the formation of a zygote. Zygote thereafter produces a multicellular, well-differentiated sporophyte, which is a dominant phase of the pteridophytes. In majority of the pteridophytes, all the spores are of similar kind. Such plants are called homosporous. Genera like Selaginella and Salvinia, which produces two kinds of spores, that is macro, large, and micro, small spores, are known as heterosporous. The megaspores and microspores germinate and gives rise to female and male gametophyte respectively. The female gametophyte in these plants are retained on the parent sporophyte for variable periods. The development of the zygote into young embryos takes place within the female gametophyte. This event is precursor to the seed habit, considered an important step in evolution. The pteridophytes are further classified into four classes: Psilopsida, example Psilotum; Lycopsida, example Selaginella, Lycopodium; Sphenopsida, example Equisetum; and Pteropsida, example Dryopteris, Pteris and Adiantum. Next is gymnosperm. The gymnosperm, gymnos means naked, sperma means seed, are the plant in which the ovules are not enclosed by an ovary wall and remain exposed both before and after fertilization. The seeds that develop post fertilization are not covered, that is, are naked. Gymnosperm include medium sized trees or tall trees and shrubs. One of the gymnosperm, the giant redwood trees, that is sequoia, is one of the tallest tree species. The roots are generally tap roots. Roots in some genera have fungal association in the form of mycorrhiza, that is in Pinus, while in some others like Cycas, small specialized root called as corallide roots are associated with the nitrogen fixing cyanobacteria. The stem are unbranched like in the case of Cycas or branched as in the case of Pinus and Cedrus. The leaves may be simple or compound. In Cycas, the pinnate leaves persist for a few years. The leaves in gymnosperm are well adapted to withstand extremes of temperature, humidity and wind. In conifers, the needle-like leaves reduces the surface area. Their thick cuticle and sunken stomata also helps to reduce water loss. The gymnosperm are heterosporous. They produce haploid microspores and megaspores. The two kinds of spores are produced within the sporangia that are borne on the sporophylls, which are arranged spirally along an axis to form a lax or compact strobili or cones. The strobili bearing microsporophylls and microsporangia are called microsporangiate or male strobili. The microspores develop into a male gametophytic generation which is highly reduced and is confined to only a limited number of cells. This reduced gametophyte is called a pollen grain. The development of pollen grains takes place within the microsporangia. The cones bearing megasporophylls with the ovules or megasporangia are called macrosporangiate or female strobili. The male or female cones or strobili may be born on the same tree as in the case of Pinus. However, in Cycas, male cones and megasporophylls are born on different trees. The megaspore mother cell is differentiated from one of the cells of the nucleus. The nucleus is protected by envelope and the composite structure is called an ovule. The ovule are born on the megasporophylls which may be clustered to form the female cones. The megaspore mother cell divides meiotically to form 4 megaspores. One of the megaspores enclosed within the megasporangium, that is nucleus, develops into a multicellular female gametophyte that bear 2 or more archegonia or female sex organs. The multicellular female gametophyte is also retained within the megasporangium. Unlike bryophytes and pteridophytes, in gymnosperm, the male and the female gametophytes do not have an independent free-living existence. They remain within the sporangia retained on the sporophyte. The pollen grain is released from the microsporangium. They are carried in air current and come in contact with the opening of the ovules born on the megasporophyll. The pollen tube carrying the male gametes grow towards the archegonia in the ovule and discharge their content near the mouth of the archegonia. Following fertilization, zygote develops into an embryo and the ovule into seed. These seeds are not covered. Next is angiosperm. Unlike the gymnosperm, where the ovules are naked, in angiosperm or the flowering plants, the pollen grains and the ovules are developed in a specialized structure called flowers. In angiosperm, the seeds are enclosed by fruits. The angiosperms are an exceptionally large group of plants occurring in wide range of habitat. They range in size from tiny, almost microscopic wolfia to tall trees of eucalyptus over 100 meters. They provide us with the food, fodder, fuel, medicine, and several other commercially important products. They are divided into two classes, that is, the dicotyledons and the monocotyledons. The dicotyledons are characterized by having two cotyledons in their seeds, while the monocotyledons have only one. The male sex organs in the flower is a stamen, each stamen consisting of a slender filament with an anther at the tip. The anther, following meiosis, produces pollen grains. The female sex organs in the flower is a pistil or the carpel. Pistil consisting of an ovary enclosing one to many ovules. Within the ovules are present highly reduced female gametophyte termed embryo sac. The embryo sac formation is preceded by meiosis. Hence, each of the cells of an embryo sac is haploid. Each embryo sac has a three-celled egg apparatus, one egg cell and two synergids, three antipodal cells and two polar nuclei. The polar nuclei eventually fuse to produce a diploid secondary nucleus. Pollen grains after dispersal from the anthers are carried by wind or various other agencies to the stigma of a pistil. This is termed as pollination. The pollen grains germinate on the stigma and the resulting pollen tube grow through the tissues of stigma and style and reaches the ovule. The pollen tube enters the embryo sac where two male gametes are discharged. One of the male gametes fuses with the egg cell to form a zygote, that is syngamy. The other male gamete fuses with the diploid secondary nucleus to produce a triploid primary endosperm nucleus, that is PEN. Because of the involvement of two fusion, this event is termed as double fertilization, an event unique to angiosperms. The zygote develops into an embryo with one or two cotyledons and the PEN develops into endosperm which provide nourishment to the developing embryo. The synergids and antipodals degenerate after fertilization. During these events, the ovules develops into seed and the ovaries develops into fruit. The life cycle of an angiosperm is shown in the figure. Plant life cycle and alternation of generation. In plants, both haploid and diploid cells can divide by mitosis. This ability leads to the formation of different plant bodies, that is haploid and diploid. The haploid plant body produces gamete by mitosis. This plant body represents a gametophyte. Following fertilization, the zygote also divides by mitosis to produce a diploid sporophytic plant body. Haploid spores are produced by this plant body by meiosis. These in turn divides by mitosis to form a haploid plant body once again. Thus, during the life cycle of any sexually reproducing plant, there is an alternation of generation between the gamete producing haploid gametophyte and spore producing diploid sporophyte. However, different plant groups as well as individual representing them differs in the following pattern. First one is sporophytic generation is represented only by the one-celled zygote. There are no free-living sporophytes. Meiosis in the zygote results in the formation of haploid spore. The haploid spores divide mitotically and forms the gametophyte. The dominant photosynthetic phase in such plant is the free-living gametophyte. This kind of life cycle is termed as haplontic. Many algae such as volvox, Spirogyra and some species of Chlamydomonas represents this pattern. Second one, on the other extreme is a type wherein the diploid sporophyte is a dominant photosynthetic independent phase of the plant. The gametophytic phase is represented by the single to few celled haploid gametophyte. This kind of life cycle is termed as diplontic. All seeds bearing plants, that is gymnosperm and angiosperm, follow this pattern. Third one, bryophytes and pteridophytes interestingly exhibits an intermediate condition, that is haplodiplontic. Both phases are multicellular. However, they differ in their dominant phase. A dominant independent photosynthetic thalloid or erect phase is represented by a haploid gametophyte, and it alternates with the short-lived multicellular sporophyte totally or partially, depends on the gametophyte for its anchorage and nutrition. All bryophytes represents this pattern. The diploid sporophyte is represented by a dominant, independent, photosynthetic vascular plant body. It alternates with the multicellular sporophytic or autotrophic, independent but short-lived haploid gametophyte. Such a pattern is known as haplodiplontic life cycle. All pteridophytes exhibit this pattern. Interestingly, while most algal genera are haplontic, some of them such as Ectocarpus, Polysiphonia, kelps are haplodiplontic. Fucus and alga is diplontic.